Genetics is the study of heredity. It's a way of explaining how living things, plants and animals, inherit certain characteristics from their parents. In some ways, we will be like our parents. In others, very different. The ideas of genetics can help us understand how changes occur from one generation to the next, or over thousands of years. These palms are all from the same group of plants, but changes over the years now make one species look very different from another. These changes occur inside the cells of living things. Cells can be seen best in thin slices of tissue. The cells may need to be stained in order to see the details. The cells of living things can appear very different depending on what they're there to do. This is what the cells in a leaf can look like. Small cells line the edge of the leaf, with longer, larger cells behind them. A common element in living cells is a darker area called the nucleus. And this is what the nucleus looks like with the magnification of an electron microscope. Inside that nucleus are the chemicals that we now believe control life and govern heredity. "100 years ago, Johann Miescher extracted an acid from the nucleus of cells. Later that century, Albrecht Kossel found 4 complicated compounds inside that acid. Nowadays, we know that those compounds form part of all living cells. They're part of DNA, deoxyribonucleic acid. These scientists are extracting DNA from the nucleus of onion cells. They're using a mixture of salt and detergent to break down the membranes of the onion cells. A liquidizer breaks down the cell walls even further. The liquid contains the DNA. An enzyme separates the DNA from other compounds. DNA will not dissolve in cold alcohol. It appears as a white layer in the tube. Early this century, Phoebus Levene discovered that DNA contained sugar molecules. But it wasn't until 1953 that Francis Crick and James Watson worked out how this huge DNA molecule fitted together. Crick and Watson depended on the work of other scientists, including Rosalind Franklin. Their model showed the 4 compounds: adenine, thymine, cytosine, and guanine, held together inside a long, strong ribbon of phosphate and sugar. The whole molecule was twisted like a helix. The DNA molecule had the ability to split open and to duplicate itself. These chromosomes are formed from DNA and protein. When a cell divides to make two cells, the chromosomes also duplicate. It's the DNA in the cells that controls the reactions taking place in living organisms. Genetic information in pollen and in the eggs of plants can come together to produce the seed for new plants. The new seed will contain genetic information from both parents. The genetic information is carried in the chromosomes, one set from each parent. There are other ways of bringing together genetic information. Flies in these cages carry pollen from one plant to another. The cages are sealed to prevent pollination by other plants whose characteristics will be unknown. The seeds produced within the cages will have some of the features carried on the chromosomes from the male parent and some from the female parent. Tomato seeds contain the genetic information that could produce a new tomato plant. Bean seeds will produce bean plants that combine some of the qualities of each of the parents. Different varieties will have different genetic information to pass on, and there will be differences even within the same variety of bean. The new plants produced by bringing together the genes of the parents will have properties in common, yet still show differences.
This whole tunnel is practically full of just one species, Cucurbita pepo. I call them squashes.
Oh, that's better.
People call them different things. Pumpkins, marrows, squashes, courgettes, zucchini, all one species, all quite able to interbreed with one another, and yet tremendous differences between different varieties. Look, I'll show you. Okay, look at this one here. Now, that's a Greek marrow.
Yeah.
It's long, smooth, and cream-coloured.
Right.
Okay, right next to it here is another one called sucrette, same species, but it's round, it's deep gold-coloured, and it's got a warty surface.
And looking completely different.
Completely different look. This one is...
Yeah.
You can see another one. That's also a Chinese pumpkin. So here you've got diversity within the variety, and this variety is different from all the other varieties. Same species, though. They can all interbreed.
These bean seeds are a store of genetic information, information that could be used to produce a particular variety of bean plant. What that plant will really look like will depend on its heredity and the conditions under which it is grown. If you're like me and have spent most of your life living in towns and cities, it's easy to believe that all cows, all sheep, all goats look pretty much the same, but not so. Here on this farm in the Cotswolds is an incredible variety of animals which have been bred at different times by different farmers.
These are Soay sheep. They're the ancient ancestors of all the sheep that are alive today in the world. They've survived by being isolated on a tiny island off the northeast coast of Scotland, and they're very, very primitive in every way. They shed their wool just like wild animals. They're naturally short-tailed, so there's no worry of getting mucky. They lamb quite naturally on their own without any help. Basically, they're survivors, island survivors. These very primitive sheep, even though they all look very similar, each one is genetically slightly different. So there are paler ones and darker ones and bigger ones and slightly smaller ones. An early man would have taken those animals which fitted his idea of a better animal and used those in his breeding program. So in effect, he would have eaten the ram lambs that were a bit too small and used the biggest and strongest ones to be the fathers of the next generation of lambs. And gradually, over time, he'd shifted the genetic makeup of the population. The Cotswold is a very good example of the extreme that man can take a breed to. They're very, very big, probably 3 or 4 times the weight of a Soay. They produce 10 or 15 kilos of wool per sheep, which has to be shorn off. It isn't shed, so we can choose when to harvest it. It's a white wool, so that it'll take dye. They produce 2 lambs instead of 1. But there is a payoff. In order to survive, they need man to feed them, to house them in the winter, and to help them have their lambs.
These are the descendants of the earliest wild pig. The piglets are striped for camouflage in the forests that were their home. Modern pigs have been bred to be larger and to have larger numbers of piglets. It's a process of selection, choosing those pigs that have the most meat and the largest litters. The properties of a plant are determined by its genes, the structure of the DNA in the cells. If you can work out which gene is responsible for which property, then that gene can be added directly to an egg. It's a process called genetic engineering. These letters represent a tiny part of the length of DNA in the cells of a cabbage. That length of DNA, the gene, may give those cabbages a useful property. Maybe the cabbages are disease resistant. One way of carrying that property from one plant variety to another is by bringing together the pollen of that one plant with the eggs of another. It's a slow process that requires many years and many generations of plants. Genetic engineering identifies that one gene, isolates it, and copies it. That gene, with its special properties, can be added directly into the plant's egg.